So, hello dear students. Today our first lecture of nanotoxicology course. I hope everyone is here, but probably not. Anyway, we need to proceed. So today we were talking about a bit about synthesis of nanoparticles, not about every synthesis, but at least about some that are very that can affect toxicity of nanoparticles. So there's a lot of them basically, but we need to discuss at least a part. But before we start, I need to remind you to fill a table with your objects of study, because not everyone still made this, and this is needed for me to give you a topic for a presentation. I think a bit about our schedule, and I think it's maybe too little time for you to present to make a presentation next Tuesday, so probably we will transfer this to next Friday. I mean this session from Tuesday to Friday, and starting from Friday we will make a presentation, not minimal 3 people but 4 people. So 4 people here, 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 and here. During this presentation, please let me know if there's 4 people to volunteer to present their topics on next Friday, so in a week. If no one wants to volunteer today, after this lecture, I will choose 4 people randomly by myself. So if someone wants to be a hero, there's time for you, and please let me know in chat. So I hope everything is clear and we can start. Okay, Anastasia and Alexandra. Okay. Not everything is so good with my voice today, but I hope I will succeed in this presentation. Let's proceed to synthesis routes. So it like maybe 3 main synthesis routes, and according to results of your introductory test, not every one of you are familiar with these routes, and this is maybe a bit more detailed table version of the table I presented you in previous lecture. So basically it's 3 main synthesis routes according to, well, let's say what we are using for basically to produce your particles. It's a physical, some physical devices and physical methods, chemical methods, also called wet synthesis, and biological methods, also called green synthesis. So chemical methods is historically the first one, and there are a lot of them to discuss. It's sol gel methods, it's evaporation, different colloidal methods, including reduction methods, and spray pyrolysis. So pyrolysis also can be used to produce particles. And all of these methods usually involve different solvents, and usually occurs in different not water, but in different liquid media. So they mostly involve different salvation of different precursors and occurs in wet conditions, so they call it a wet chemistry. But not always, of course. And if we're looking on physical methods, there's a big group. Let me turn laser pointer. So it's a big group of methods involving different laser methods like laser ablation, laser pyrolysis, and also sputtering. So sputtering, it's the same as maybe you used a sputter coater in our lab, sputter coater to provide a coating for scanning electron microscope. So it's just a metal plate under very high voltage, and some particles can not even atoms, but like a liquid metal drops that can produce by a high voltage. So it's just a heated metal plate which can produce very small drops of metals, which can form nanoparticles. So laser ablation, laser pyrolysis is almost the same, but the high temperature obtained not by high voltage, by just a focused laser beam. And of course there's some kind of other physical evaporation methods, but among physical methods there are also mechanical methods like mechanical modeling. So you can just use some physical strengths to mile to destroy some particles, large particles into small particles. But it is very hard to obtain some nanoparticles using just mechanical mining. And among these methods can be we can include like sonification, so using of ultrasound in mechanical mining. So our magnetite produced in our laboratory, basically some kind of ultrasound assistant can be called like mechanical methods. And if we look on green synthesis, it's not because we using, for example, green colored reagents or something like this. It's because we're using something occurred in nature. And green synthesis was basically called green because they was using some plants like leaves or roots or something like this to produce particles. But now green synthesis is everything that involved using of bacteria, fungi, yeast, extracts from leaves and so on, and even whole cells. So again, according to the result of introduction test, not all of you familiar with the green synthesis method, so we'll discuss it today. Well, if we're talking about synthesis methods, there's a lot of particles that can be obtained using these methods, and they can be divided into two big groups. So these two groups are obviously organic and inorganic particles. Of course, there's not so strong division on organic and inorganic because on the border between these particles some kind and some type of hybrid particles, and this part of hybrid particles may be even bigger than just strictly inorganic or organic particles. But if we're talking about terminology and we were talking about hybrid particles, as for me, only for me, hybrid particles is something that consisted of organic and inorganic parts. So, for example, gold nanoparticles covered in polymeric micelles, in polymeric molecules. It can be called hybrid. But for some other scientists, for some other researchers, for example, gold nanoparticles covered in silica shell also can be called hybrid because it's like a hybrid between gold and silica. But for me, it's like a core shell inorganic nanoparticles because both of the components are still inorganic. But it was, everything is just a terminology, and you need to pay attention when you are reading some papers, what is called hybrid and why it's called hybrid. And of course, since we are talking about different kind of toxicity, we need to discuss advantages and disadvantages of both of these types concerning bioapplications. If we are talking about organic particles, the most important advantage is biocompatibility, because all of this is made basically of organic molecules which are naturally present in our organisms, so like proteins or lipids or polymers and, well, maybe not dendrimers, but still a lot of these molecules are naturally occurs in our organism, and that's why these particles have an excellent biocompatibility. Of course, you can precise target these particles, especially in cases when you have some conjugates or you have some antibodies on top of these polymeric micelles, for example. And antibodies and well, not only antibodies, but different natural ways to accumulate these particles are also present. So when you synthesize these particles, you usually use less hazardous reagents and synthesis methods. So it's like so-called mild chemistry, soft chemistry in some way. So basically, you do not need some very hazardous solvents to produce these polymer or protein particles. And if you look on all of these examples, you will definitely see that all of these particles have a very big surface area, especially dendrimers. So if they have a big surface area, it's a big area for different functionalizations. So you can add different molecules to the surface of these particles, and surface area is very important when we're talking about different drug delivery techniques. And one of the most important also, last here but not least among the advantages, longer circulation in blood. So some of these particles have a really long circulation in blood, and we will discuss it maybe in our third lecture about functionalization. And if we talk about disadvantages, well, still there are some disadvantages, obviously. Everything cannot be so good. And the first disadvantage, it's a disruption of cell metabolism. So if the cell will uptake a lot, for example, of liposomes or protein nanoparticles, it definitely will twist some of intercellular metabolism pathways. And if it can be compared to a human, if you drink or eat a lot, you will be feeling like ill, and the same as cell. So if the cell will uptake a lot of polymer nanoparticles, for example, it can cause some alteration in cell metabolism. So maybe not very serious, maybe the cell will survive this, but definitely it can cause some disruption. And of course, despite this, all of these particles are biocompatible, still they can cause immune response in organism of human or animal. And maybe it's not a big disadvantage for some types of particles, like for liposomes, this disadvantage already overcome, but it's for some of these, for polymeric, for dendrimers particles, it's not that so easy to produce a large bunch of this particle, for example 1 liter of dendrimer particles or something like this. So it's basically not very well scalable synthesis approaches. But if we switch into inorganic particles, there's also a lot of advantages. For example, bioinertness. Just to compare biocompatibility and bioinertness, it's completely different things. Because if organic particles can be used as building blocks for organism, for example, liposomes can be used for building of cell membranes. For example, gold nanoparticles, as gold is a bioinert metal, they can be just stored in some cells, for example, in kidneys for months, for years, without any significant side effects. So bioinertness of some of the particles are very important. And since all of these are, well, basically metals or non-metals, but still they have some ability to be manipulated by physical stimuli. So gold nanoparticles, for example, can be not maybe manipulated, but you can obtain some, can manipulate, for example, temperature of these particles by illuminating it with light with a particular wavelength. Or for example, you can manipulate magnetic particles with magnetic field. And based on this advantage, you also can use them, use their unique optical properties. For example, for CERS, surface enhanced Raman scattering, it mainly connected with gold nanoparticles as well as surface plasmon resonance. So some of the particles have really unique optical properties that you can use. Again, last not least, the active labeling. You also can label, for example, magnetic nanoparticles with some iron ions, iron labels, and use them for visualization maybe. Of course, again, disadvantages. And the first and the most important disadvantage is the generation of reactive oxygen species. So these particles are mainly not able to produce different ions, but these particles can. So magnetic particles can produce ferrous ions, iron ions, gold ions, Well, silicon nanoparticles, it's oxide, so it will produce less amount of ions, but other different particles, for example, quantum dots can produce cadmium ions and so on. So we will talk about ROS a bit later. And of course, immune response much stronger in comparison with the immune response from organic particles. And because this immune response is much stronger, it will definitely cause inflammation in tissues and organs. And this is a very important concern when we're talking about inorganic nanoparticles. And also accumulation in mononuclear phagocytic system. So these organs like liver, kidneys, spleen, and so on. And these particles strongly accumulated liver mainly. Lungs are not part of MPS system, but still lungs also accumulate a lot of particles. And also it is important that most of these particles have a rapid clearance profiles. So these particles can be really and fast eliminated from the organism. For example, as I already said in previous lecture, particles with a size less than 5 nanometers will go straight to the kidneys and eliminate maybe within an hour. So it is also very important. And now we need to move to the synthesis methods itself. So we will start from inorganic nanoparticles, and the first method to discuss is solvothermal synthesis. So solvothermal synthesis is a very common way to produce a lot of different particles. And these methods also involved hydrothermal synthesis when you used water as a solvent, but in general you can use any solvents to produce these particles. I think most of you saw how to produce particles using solvothermal synthesis. You need some stainless steel autoclave and some Teflon inliner. And you place some of your precursors dissolved in some solvent, and you tie this with stainless lead and place in some heater. So solvothermal synthesis, as you can see, it's thermal synthesis. So it used some temperatures above, basically above 100 degrees, and some elevated pressure. What's so important about this synthesis is it can use to produce crystalline particles, not just crystalline, nanocrystalline particles, because when you heat your precursors and then slowly cool these precursors, they undergoes crystallization to particles. And using this technique, you can produce metal oxides, different gallium alloys, carbon nanoparticles, quantum dots, and so on. But the most important thing is emphasized here, it's a crystallization. So when you're talking about nanoparticles, in most papers you will never see additional investigation connected with impact of crystallinity on toxicity of particles. So for most of the scientists, it doesn't matter amorphous or crystalline, important just maybe shape, size, and composition. But it's not true, because if we look on, for example, titanium dioxide particles, which is one of the most produced in our world, there's a lot of different crystal modification of these particles. So it can be like anatase or tile, and also it can be in amorphous state, and there's some kind of other states. And there's a very nice phrase from this article: how meaningful are the result of nanotoxicity studies in the absence of adequate material characterization. So when you're reading, for example, about titanium dioxide nanotoxicity or just toxicity, you will probably see a very short phrases about what was the main phase of this titanium dioxide. But if we look on this histogram, we will see that different kind of crystal structure have different impact on toxicity of these particles. Now, maybe the difference not that high for some concentration, for example, like let's see, amorphous phase and crystalline phase, like 100% rutile, but still there's some difference, maybe like 20%. So if we are talking about crystallinity of the particles obtained by solvothermal synthesis, it's also important to assess dependence of nanotoxicity from crystalline phase. And what's so important about crystallinity, how this crystallinity can affect nanotoxicity. So if we look, for example, on this paper, there's a lot of zinc oxide particles with different structure, and you definitely can see that this zinc oxide particle have some crystal structure because it's diffraction on transmission electron microscope, and you can see crystal lattice here. And based on different crystal structure of the same zinc oxide particles, you see that zinc oxide, let's say called here A and B, have different toxicity profiles. And also here, so it's just a different cell cultures, and it's also important because when you're testing your, for example, nanoparticles or other formulation, it's also important to pick a right particle for testing because, for example, for one, not particle, cell cultures for testing, because for these cell cultures, these zinc oxide particles are not toxic, but for these cell cultures, they're toxic. And also there are difference between different formulation of this zinc oxide based on crystal structure. So if we're going a bit further, it's also important to look on biodistribution for different organs. So if there is a lot of parameters that can be assessed when we are talking about crystallinity of these particles, crystallinity and assessment in human organism or animal organism. Because, for example, on this plot, there's a lot of them, but you can see that some of the particles, it's quartz particles, so quartz, different kind of quartz particles. So most of them, for example, in this particular paper, most of them are non-toxic, but just one formulation was toxic due to its crystal structure. And also you can observe this difference using some fluorescent staining, but we will not talk about this in details because it's not very interesting probably for you. But one thing you need to memorize is that crystal structure are important when you consider a nanotoxicity of particles. So for most of the scientists it's not important, but you should be better than this, and you should know about this. The next methods used for production of different particles, mainly gold nanoparticles, silver nanoparticles, and so on. It's a chemical reduction methods. So when we are talking about chemical reduction, it's a long history of chemical reduction, for example, of gold. So the first methods were described even by well, maybe in 19th century, but the most usable one is a Tukevich method, and it further was adjusted by other scientists, and reduction by Skobe case, it was implemented maybe like 30 years ago, and now a lot of papers devoted to green synthesis of gold nanoparticles. And if you are interested, you can just read this very recent paper about different methods for producing of gold nanoparticles. But when we're talking about different reduction methods, it's one very important thing you need to know about particles which produced by chemical reduction. To provide a stability for these particles, you need to add something to their surface so they wouldn't aggregate. And when these particles produced by reduction, there's electric double layer on the surface of each particle, and these particles called usually electrostatic stabilized, so electric stabilized. And This stabilization is not very good because if you add something in your media to your particles, it will definitely cause some aggregation. And also, when we're talking about chemical reduction methods, it's a lot of options to manipulate the size, the shape, and other parameters, also electrostatic stabilization of these particles by varying ratio of precursors used for the synthesis. And despite these methods already like 50, 60, 70 years was developed a long ago, there's still a lot of research papers published even in 218. Well, since this paper is from previous year, there's not much data available, but still reduction methods are still used and well, very successfully used for producing of still gold nanoparticles. And let's talk about a bit precisely in details about stabilization of these particles. So when the nanoparticle, on example of gold nanoparticles, when gold nanoparticles produced, it basically consisted from gold ions, iron 3 plus, and on the surface of these particles, in case of, for example, reduction by sodium citrate, there's a lot of hydroxyl groups, and this is electrostatic stabilization basically. But there's also other methods to produce different type of stabilization, and also you can add some functional groups later after the synthesis, and this will result in formation of different type of electrostatic stabilization. But still this is electrostatic stabilization, so it's just charged molecules on the surface of your particles, and this is just electrostatic repellent between your particles. But why all of this important when we're talking about nanotoxicity? Well, it is important because surface charge of nanoparticles strongly affects cell uptake. If you probably know, probably not, cell membrane is negatively charged. And because this cell membrane is negatively charged, of course, if your particles also negatively charged, they will have low affinity, and this will affect endocytosis strongly, and these nanoparticles will less frequently enter your cells. In case you have positively charged nanoparticles, endocytosis will go much faster because of high affinity of these particles to a cell membrane. And definitely it can be some other functional groups on the surface of your particles, but basically negatively charged nanoparticles are very good for a longer circulation in blood, while positively charged nanoparticles are mostly used for intercellular uptake. So it's very important concerns about using differently stabilized particles. And also, if we use some other kind of stabilization for your particles based on charge, it's important to note that intercellular weight of these particles is also different, because, for example, if you coat your particles with dextrane gives like a zero charge, so it's neither negatively or positively charged. They will accumulate in cytoplasm, and this is will basically will be non-toxic. But if you have some positive or negative charge, they will accumulate in lysosomes because they will undergo some endocytosis. And if they damage these lysosomes, it will definitely cause some disruptance in cell cytoplasm pH level, and this level will definitely lead to some cell death, maybe even some necrosis, apoptosis, and inflammations. So it also important. Also important to know what's on the surface of your particles, not only charge but also coating. Well, charge are not just charge, of course, because charge connected with another aspect of reduction produced particles called zeta-potential. So zeta-potential is not very easy term to discuss, but probably again, many of you heard about zeta-potential and measured zeta-potential on our photocore device. So when you use, for example, gold nanoparticles obtained by citrate reduction, there will be a lot of citrate molecules on the surface of your gold particles. And this particle will be called like citrate gold nanoparticle, citrate stabilized. But you can use some displacement procedure, for example, with some thiol groups, and it will be like thiol gold nanoparticles. Now, but still, you can see there's a lot of surface groups which are used to stabilize this, and based on these groups, this particle will have some surface charge, and of course, they will have some potential. But what is zeta-potential? Zeta-potential, if you have a particle, you will definitely have some Stern layer. So Stern layer is something, is ions absorbed on the surface of your particles. So this layer has the highest potential among all other layers. And of course, there will be some slip and play, so your particle moving in the media. And there is some diffuse layer, so it's ions that came to the surface of your particles or came in, came out, and the potential, so the difference in voltage between these two planes called zeta-potential. So it's not a surface charge, it's just some kind of measure of your particles, and this measure is very important because zeta-potential, well, not directly, but still it's a measure of stabilization of particles. And if we look on different zeta-potential, which is measured in millivolts, if we look on different zeta-potential, usually for different metal oxides, most of us working with metal oxides or just metal particles, for most of the particles to provide a stability of those colloidal solutions, you need a zeta-potential more than 30-40 millivolts, or for in case of metal those even higher, like 70 millivolts. And it doesn't matter what the charge, so it can be both negative or positive charge. Now, for polymer or emulsions, it can be a bit lower, but still it should be like 20 at least to produce aggregational stability. But if we look on high or low zeta-potential, still we are talking about nanotoxicity, we need to discuss something connected with nanotoxicity, how the zeta-potential can affect this toxicity. So if we have a high zeta-potential, the particles will definitely weakly dispersed, so they will not sedimentate, so they will stay in solution, and sedimentation stability will be very good. So the viscosity will be low, and most of the particles will remain in liquid in separate state. But if that potential is low, so opposite case, the sedimentation stability still be very good, but not because nanoparticles stay separate and they are very light and there's some brown moving, but because viscosity will increase, and the increased viscosity will affect stabilization of these particles, so sedimentation stabilization of these particles. But in some case of average zeta-potential, particles will definitely aggregate, the viscosity will be not so high, and these particles will sedimentate. And if you add these particles, for example, to your cell cultures for testing, these particles will sedimentate on the surface of your cell cultures, of your cells. And also a couple of words about zeta-potential and the importance of this zeta-potential. It's isoelectric point. So isoelectric point very important for different proteins and other molecules. So some of them particles have isoelectric point, can provide isoelectric point for absorbed proteins. So I believe Andrei in his course of bioapplication of some particles should talk about this. Now, for example, if you have zeta-potential of your particles which meets isoelectric point of protein, this protein will not, it will not absorb on the surface. So it's basically a very good strategy to provide additional stability for your particles in case of using in blood, for example. So this is a very tricky and very let's say narrow window for particles, and it's not very easy to control at that potential, but still this option exists. So if you want to produce add additional stability to your particles, it is better to give it some zeta-potential as for the electric point of most abundant proteins, for example albumin and globulin. And again, a bit, well, it's just is electric points of different proteins which can be found in your blood, for example. So why that potential is so important again? When that potential can be also used not just to assess stability of your particles, but also to measure or maybe to assess structure of your nanoformulations, microformulation. So depending on structure of these layers on the surface of your particles, they can be like bare particles, like maybe magnetic particles, magnetite or gold particles, and the dependence of that potential from distance will be like exponential. But if you have some absorbed molecules on the surface of your particles, the curve will have like plateau, and next it will be like exponential decrease. In case of this is in case of thick charged layer. In case of thin charged layer, this plateau will be much smaller than here. Well, if you want to read a bit about that potential, because it's a very powerful instrument for assessing your particles, you can check this paper. So it's a very nice one to say. And well, but that potential can be used not only to assess charge and other things, but it also can be used to assess ionic strength of your solution, viscosity of your solution, concentration of particles, their size, composition, and charge density. And there's a lot of things that can be assessed using just that zeta-potential, but we need to move to what I want to tell about why zeta-potential is very important. Because when we are talking about particles, we are I'm not sure about you, but at least for me, when I'm using some particles and I want to assess their toxicity using some cell cultures, for me, basically, it's not very important will they aggregate or not. So almost all particles, even if they are very stable in water, for example, they will definitely aggregate when you add them to culture media, because culture media have a very high ionic strength. There's a lot of ions, divalent ions like potassium, magnesium, sodium, and so on. And these ions will decrease electrostatic stabilization. They will definitely eliminate any electrostatic stabilization features of these particles. And when you're using just electrostatic stabilized particles, which produced by reduction methods, basically, they will definitely aggregate in culture media. So it's true for gold particles, for silver particles, or for magnetite particles. But what about aggregation impact on nanotoxicity? If we have, for example, very well dispersed particles in your media, you will definitely see no nanoparticles via optical microscope because they are very small. But in case of poorly stabilized particles, they will sedimentate, aggregate, and you will start see some particles which here like presented here like black dots. But what about non-aggregated and aggregated toxicity? Well, aggregated particles have a bigger toxicity, so cell viability will decrease. maybe not very high. So as you can see, it like 90%, it like 60%. So the difference is like 30% only, not like 90 and 0. Yes. But still, aggregated particles have a stronger impact on this on your cells because of just one feature. If you look on scanning electron microscope images, you will see that aggregated particles strongly interact with the cells and mostly with the cell's membrane. So it's just physical damage. So these particles in form of aggregates can affect structural stability of cell membranes, and in some cases this damage can be fatal. So if we are talking about in vitro testing, you also always need to bear in mind that if your particles aggregated, they will definitely affect viability of your cells. So it is better to provide some additional stability by, I don't know, coating with PEG or coating with dextrains or something like this. And this is connected with not only with chemical reduction methods, so using, for example, some citric acid or something like this, but if we're talking about laser ablation. So if we're talking about laser ablation, what are the mechanism of laser ablation? You have some gold plate, for example, and you use laser to produce some particles. And if you look on these still gold nanoparticles, and if you look on these gold nanoparticles, they looks very well, and they have a plasmon resonance peak, and everything is good with this. But what about stabilization? And what about stability and quality of these particles? So if we're looking on what are the drawbacks of laser ablation, of course you can produce this in a solution still containing some additional polymers. So if you add some polymers in this solution, the obtained particles will immediately cover in these polymers proteins, and they will be stabilized by these polymers. And this is already not a chemical, this is already not an electrostatical stabilization. So maybe it's a bit better than in case of chemical reduction. But if we're talking about laser ablation, it's very hard to control polydispersity of the particles. If we look on distribution of these particles, distribution looks very good. But if you look on 10 pictures, well, there's a lot of big particles, a lot of small particles, and all the particles have completely different sizes. Of course, there's some ways to improve this distribution, but most of the physical methods, they produce poorly distributed nanoparticles in terms of the sizes. In some cases, especially in cases of optical properties, this is not very good, because very wide polydispersity, very wide distribution will result in a wide peak. So for poorly distributed particles, plasmon resonance will be maybe like this one. So it's not very good for optical properties. And if we look, it's just very nice pictures. It's a hole formed by laser following laser ablation, synthesis of nanoparticles. So it's just a very small hole. The size of this hole maybe like 1 micron, 2 microns. So just looks very nice. But what are the alternative for the reduction methods, reduction synthesis methods for example, gold nanoparticles? It's a green synthesis. So green synthesis can involve a lot of different reagents, but basically it's still a chemical synthesis, just using some extracts from leaves, for example. And you still need some gold salt, some gold precursors to produce these particles, because as far as I know, there is no such a synthesis when you use one extract and second extract and you obtain gold nanoparticles. It's just gold from nothing. But for now, this extract's used because it's a bit more, a bit better for environment than using some, I don't know, other harsh reagents. In case of this green synthesis, this is important that you can produce a really large amount of these particles. So on this picture, someone holding a whole plate of particles. This is not just the Earth, it's a bunch of particles, maybe nanoparticles in some way, produced by green synthesis. So in some ways, the synthesis methods are quite scalable because, for example, a leaf extracts can be obtained in large amount. You can use a whole tree to produce this extracts, and the question only when where you will obtain so many gold salt. But in case of green synthesis, it is very hard to produce good particles in terms of shapes. For example, I think every one of you knows Sansevieria is a common home plant, and you can use the leaves to produce leaf extracts. And use chloruric acid and obtain some gold nanoparticles, which, for example, in this paper were used to degrade some organic dyes. I'm not sure why this is so important to degrade, for example, Acadine orange or Congo red, but still it's just a research paper. But if you look on this particular TEM image, you will see that the particles have very poor size and form distribution. So shape of these particles like triangular, like ellipsoid, spherical, and they have a very uneven surface. And if you look on plasmon resonance peaks, they are very wide. So these particles are not very good for different optical experiments or optical applications. But for some maybe degradation of organic dyes somewhere in lakes, oceans, I don't know, maybe these particles is good for these purposes. But another alternative to the green synthesis, well, it's still green synthesis, but not with extracts, but with whole cells. So yes, indeed, some nanoparticles can be produced by a whole cell, and in this case, this is bacteria. It's not very interesting after all, because synthesis with bacterial cells are already common, and it's like maybe 20 years ago when the synthesis were firstly involved. But for example, using this bacteria, it's still, I believe, Escherichia coli. So if you we look on this bacteria, there's a lot of particles that were synthesized using this bacteria. It's like iron, silver, gold nanoparticles, some ferric cobalt particles, cobalt nickel particles, and cadmium particles, and all of these, look, there's a very large amount, like maybe a gram of particle. Now, all of these examples were produced by living bacteria. But not only bacteria can produce particles, because if we look, for example, on leaves on some plants, plants also can produce some nanoparticles. You just need to put a bit of silver nitrate in the water and pour this water on your plant, and after 24 hours you will able to find some silver nanoparticles in the leaves of your plant. So these particles really were formed only within 24 hours in some plants. And if you look on energy dispersive analysis, you will see that, of course, there's some cuprum, because it's cuprum mesh used to visualize these particles. But still, you can see some silver here, and probably something silver here. But not only bacteria, fungi, yeast, plants can be used to obtain particles, but also even mammalian cells can be used. And this one is human cells, and there's some particles which were obtained in human cells just using some gold-containing salts. So if you want to read a bit about biosynthesis of inorganic particles using eukaryotic cells, you can check this review. Yes, of course, it's molecules, it's MDPI, but it's very recent article. And there really some very nice research is described there. So almost any of cells can produce particles. I even once heard about in vivo synthesis of gold nanoparticles. Researchers, sadly, I can't find this paper, because researchers just take a gold chloroauric acid, inject it in a tail vein of a mice, and next they find some gold nanoclusters in the cells of a liver. So mice, I mean cell of mice, cells of mice liver produce some nanoclusters, gold nanocluster by its own, using just, I don't know, some reductases or something like this, following just one intravenous injection of chloroauric acid. So even a whole organism can synthesize nanoparticle by its own. Well, so we need to move further to the next type of synthesis, and this type of synthesis called microemulsion synthesis. So if you look on microemulsions, there's a lot of different subtype of synthesis of these microemulsions, but basically the scheme is something like this one. You have some oil and you have some surfactants, and you have some reagents A in this emulsion, yeah. Then you have also some, again, oil, some reactant B, and again surfactants. You mix this to mix these two mixtures so they can form a solid mixture, and in case when A and B meet together, they will form some particles, some some new components, and because these particles, these micelles, let's call them micelles, they're very small, the method's called microemulsions. But if you look on this microemulsion, for example, again, another one example, oil phase here, aqueous phase here, still you can add them, add some reactant B, reactant A, so mix together, and you will obtain some new components, some new particles, or something like this one. But all of these pictures, all of these methods have one common feature, and this common feature are not very healthy for cells. And I give you some chance to put something in chat, so please tell me what this feature about all of this synthesis, which are not very healthy for cells, just for me to rest a couple of seconds. So what this synthesis, what is the similarity between this synthesis, and what is the component of the synthesis which are not very healthy for cells? For me, it's obvious, of course, but according to results of introduction test, most of you know that this component are not very healthy for cells. It seems for me that no one is listening, probably. Well, nope. It's like 14 people here. Okay, surfactant. So surfactants always used in production of any microemulsions, and these surfactants, well, they were toxic. But if we're talking about surfactants, there's a lot of them, and there's a lot of different kind of surfactants, and you need to know, they are also called amphiphilic molecules because they have hydrophobic and hydrophilic parts. And among surfactants, there are 4 types of these surfactants exists. Well, what is surfactants? It's what you use to wash your dishes, for example. If you use a fairy bottle and look on a composition of this fairy, you basically will see at least anionic or maybe non-ionic surfactants in composition without any details of what's the type of this anionic or non-ionic surfactants. But there are also zwitterionic and cationic surfactants, and all of them have different toxicity profiles. So if you look on this one, for chemicists, it may be obvious what this surfactants called. It have some bromide, and this structure probably also very familiar, but maybe someone want to put the name of this surfactant in chat. Maybe not. Bromide, what have bromide in composition? At least half of you, I think, heard about this surfactant. No one. Well, okay. So it's ethyl-trimethyl-ammonium bromide, and this is a very common component of almost all chemical synthesis which involve formation of surface anisotropic particles. So if you want to produce some star-looking particles with some spikes or rods, for example, elongated particles, you need to use some surfactants to produce it, and also they use to produce some micelles. And these cationic surfactants, they are the most toxic ones among all of this, all of other surfactants. The next surfactants based on its toxicity, it's zwitterionic surfactants. So zwitterionic surfactants possess both of the groups, so they have cationic and anionic groups, and since they have a cationic group, they are more toxic than anionic surfactants, which possess only anionic groups. And the less toxic among all of these, it's non-ionic. So for fairy or other soaps which you use for washing your dishes, anionic, not even, well, maybe anionic, maybe non-ionic surfactants used because they are basically non-toxic. And these surfactants can form different structures in media. For example, they can form so-called micelles, they can form vesicles, reverse microemulsions, direct microemulsions, and all of these are very nice, but how these microemulsions and the structure of surfactants interconnected with toxicity? So what is the primary cause of cytotoxicity of these surfactants or amphiphilic molecules? If you look on their structure are very similar to something you can observe in the cell. So there's some kind of head and some kind of tail. And if you look, for example, maybe this looks more similar to you, so a head and a tail. And in our cells, there's a lot of such structures. It's just a bilipid layer which forms cell membrane. So cell membrane have some polar heads, non-polar tails, and this and surfactants mainly accumulate on the surface of cell membranes. So when some amount of surfactants accumulate on cell membrane, it's yet not very toxic. But when you increase concentration of surfactants, these surfactants will pass through this membrane and they will disrupt at least part of the cell membrane. And when the concentration increase even higher, there can be a situation when part of the cell membrane surrounded by these surfactant molecules, and this part of cell membrane just flow out of other cell membrane, and this will lead to a formation of a hole, and this hole cannot be repaired by a cell itself because, as you can see, surfactants shielding these molecules, lipid molecules, and this hole, it's just a hole as well as I don't know, wound in your organism. So if you make a really great hole in your skin, you will definitely die. So the same is true for cell. If you make a lot of holes in the cell membrane, the cell will definitely die, and basically will die during following necrosis process. So it will be a very fast death. And in some cases, when you have some special kind of surfactants, They can form so-called barrel-stave mechanisms. They can form channels in the cell membrane, and these channels not very good for the cell, because this also will affect ion gradients and transport through cell membrane and so on. Well, if this is just maybe a bit detailed scheme of how these surfactants affect the cell membrane structure. So if you have some micelles formed from surfactants, they just take out some lipids from a cell membrane, and so the DNA leak out and the cell will die and so on. And well, that's why the surfactants are very toxic, and the main toxic mechanism of these surfactants is a cell membrane damage. That's why when you're using some microemulsion synthesis, you need to wash your particles with cyclic washing at least 3 or 4 or 5 times until you will not see any bubbles in your washing liquid, because bubbles is a signal. It's a signal of surfactant presence in your nanoparticle solution. So microemulsions is good, but well, surfactants are not. And let's move to magnetic nanoparticles. So it's a lot of different types for synthesis of magnetic nanoparticles, and basically it's some again reduction methods using some for example ammonium solution and some precursors like iron chloride 2 and 3. And when we are talking about different methods, it's like co-precipitation methods, reverse microemulsion, again microemulsion, surfactants, and so on. And also it can you can use a thermal decomposition, well, and co-precipitation as we used in our laboratory. But among all of these methods, co-precipitation may be the most biocompatible one. You don't need to wash these particles for a very long time to get rid of, for example, surfactants. But even non-toxic is toxic. So if you look on superparamagnetic iron oxide nanoparticles called spions, they can be accumulated in the cells. It's like a healthy cells. It's not very healthy cells because it's a large amount of particles accumulated in the cell, and we will talk about maybe in lecture 4, I think, about toxicity of magnetite nanoparticles, because ferrous ions involved in a process called ferroptosis, and ferroptosis is a very important part of cell death. But when I'm talking about non-toxic is toxic, if you look, for example, it's just a recent investigation made by me about our magnetite. Many of us working with our magnetite in our lab, and if we look on this infrared spectrum, you definitely will see some peaks corresponded to magnetite, and this one is very interesting thing. So this one is traces of ammonia in I think like six time washed magnetite. So this magnetite will definitely be toxic because of these ammonia traces. As you can see, there's a lot of ammonia still in this magnetite solution. Now, so even such methods as co-precipitation need a proper washing techniques to get rid of these some traces of toxic reagents. And finally, we will move to organic particles, nanoparticles, and well, there are also some methods that will result in produce less or more toxic formulation. First one in nanoprecipitation method. So nanoprecipitation method already like maybe, I don't know, 40 years ago, it was maybe 1989. So for now, nanoprecipitation method used to produce a lot of different type of particles, and this method not very not very difficult to understand, but still why it's nano and precipitation. So you have some again some border between two phases, like oil phase and some solvent phase. And when you drop some of your reagent in this mixture, for example, you have oil here and you have solvent here, some of the solvent will diffuse and oil will diffuse, and on the border of this between these two phases, when you have, for example, polymer dissolved in your oil, the polymer will precipitate. So and varying concentration, varying, for example, syringe pump speed, and varying steering with magnetic bar, you can adjust and control this, control the size, distribution, and so on of these particles. So polymer will precipitate on the border between on the border of this emulsion. So using nanoprecipitation technique, you can obtain a lot of different kind of particles, and if you adjust these methods, you can really control a particle size, polydispersity, zeta-potential, even crystal form, stability, nucleation, growth rate, and a lot of other different parameters. But what's so special about nanoprecipitation method? So nanoprecipitation method mainly used to produce different particles from polymers, and among all of these polymers, one is very famous. So this is PLG, polylactic glycolic acid. And you can see how this particle looks. There looks very smooth, very nice, with a very nice polar dispersity. But if we look and compare the toxicity with other inorganic particles, basically, this experiment conducted on cell lines, two different cell lines, HeLa cells and CACO2 cells. Both of these cells are tumor cells, so it's cancer. And if you look on biocompatibility of PLG particles, you will see that in some concentration, of course, it's not it's statistically not very significant, the difference between these bars, but still, even PLG particles have some signs of cytotoxicity, even if this, despite the fact that PLG called one of the most biocompatible among other proteins. But this is only for these models, for these cells. And if you probably know, or maybe not, HeLa cells are not very human after all. After all of these years, it's been like maybe 60 years already, 1954, something like this one. This HeLa cells was obtained from Henrietta Lacks. So for now, these cells have like 90% of human genome. So 10% is just some kind of mutations, random mutations. So testing on HeLa cells in year 2020 are not very good choice. Well, this work was done like 10 years ago, but still, it's not the best option. But despite in this particular research, PLG particles have some good biocompatibility, like maybe 10%, 20% of cell death. If we look on other researchers, for example, using PLG particles as drug delivery for different application, if you look on PLG or PEG-coated PLG, PEG it's polyethylene glycol, so another one biocompatible protein, not protein, polymer. And PCl it's another type of biocompatible polymer. So if you look on PLG and at a very high concentration, like 200 milligrams per milliliter, I believe, you will see that PLG is not so biocompatible after all. And if you look on other, it's day 2, day 4, and day 6. Well, at day 6, all of the cell viability in all of the samples dropped like maybe 50%. But this may be not only due to cytotoxicity, but we need to check experimental protocol. This can be because of depletion of some nutrients in culture media, for example. What this research is performed to reduce PLJ toxicity. For example, what the staining for? This staining is used for so-called tunnel staining, and this tunnel staining used to detect dividing particles, and not just dividing particles, but errors in mitosis. So this, if you look on PLJ particles, you will definitely see a lot of bright dots. It means that PLJ have something to do with mitosis of particles. And to reduce, for example, toxicity of PLJ, you can use pegylated PLJ. So it's just polymer in polymer. And if you use some formulation, you will definitely see that pegylated PLJ are much more biocompatible, in no difference with control, are much more compatible in comparison with just PLJ. So even non-toxic can be toxic if you pick up a right model for testing. And we will discuss it maybe on lecture 5 or 6, that you need to rationally choose a test model for your particles. So if you have intention to find toxicity, you will definitely find some toxicity. What other methods to produce, for example, other methods to produce protein particles, polymer particles? Well, protein is still a polymer, so it's a desolvation method. So desolvation methods used to produce different kind of protein particles, for example, from albumin, human albumin or bovine albumin. And the desolvation, it's basically reducing of solvation state of proteins. So if you have some protein, enzyme still protein, and you add some ethanol in this solution, the solvation of this particle will decrease, and they will desolvate in some bigger aggregates, and you need to add some cross-linker to stabilize these particles. And this is the primary way to obtain some protein-based particles. If you do not want to use some cross-linkers, because cross-linkers are very toxic, basically it's some kind of glutaraldehyde or something like this one, you can use some method, of course it's me, some methods that we discovered previous year, without using any cross-link agents. Just add some PEG, polyethylene glycol, in your media, and this PEG will cover these particles. So no cross-linking, just a PEG shell around these particles. And this PEG shell will stabilize these particles, so they will decompose maybe for 2 days, providing a slow release. But if we're talking about different protein particles, there's another type of protein particles called nanoclusters. So looking very nice, very beautiful, and this is not a quantum dots. It's just a atom of gold surrounded by a protein. Basically it's albumin of different kinds, like bovine albumin, human albumin, and these are very nice structures that can be used as natural analogs for fluorescent staining. So just for you to know that there exists some such a kind of objects called nanoclusters, so gold nanoclusters, silver nanoclusters, cuprum nanoclusters that possess some properties of fluorescence. They are not very toxic after all, because it's just a metal ion, a very small amount of metal ions surrounded by a protein shell. So protein basically very biocompatible. But let's move to another. I believe it's like maybe. Let me just check a bit. Yeah, it's almost end of our lecture, and I have still some time. Different kind of biomimetic particles. So what is biomimetic, mimicking, call them as you want. What are so important with these particles? All of the previous example of organic particles, they involve some kind of proteins or polymers, which are, where they are very close to human nature, but still they are not from a human. Even human serum albumin is obtained from some other humans. So to produce fully biocompatible particles, you need to take your blood, separate your your albumin, and produce particles from your own albumin. And even in this case, when you produce these particles, there will be some immune response, because when you add some ethanol and use some cross-linker to these particles, the conformation of this protein will change, and this definitely will trigger some immune response in your organism. So what are the simplest way to produce some biocompatible particles? Well, we have a lot of different cells in our organism. For example, platelets. Platelets are mainly involved in formation of clots in our organism, and there's a lot of platelets in our blood. And we can cover some of the particles in platelets membranes. So it's not very hard to produce a platelet membrane. So we just need to adjust ionic strength of solution, so these platelets will just pop up, everything will go in media, and we just collect these membranes of the platelets, and then we can use these platelets to produce some of the particles, to cover some of them in membranes. And since platelet membrane have some receptors on its surface, it can be used, for example, to target these particles, naturally target these particles to wounds, to tumors, to target some cardiovascular diseases, and so on. Again, you can check this paper for some additional details. So how is this working, and why platelet-derived particles are so popular? If we look on platelets, platelets have a lot of different receptors on their surface, and these receptors can be used to target tumor cells, for example, through collagen or fibronectin, for example, receptors. They can be targeted to injured endothelial cells during a thrombus formation, clot formation, so using again fibrinogen receptor or thrombin receptor. And also platelets are involved in elimination of bacteria, and along with the neutrophils. So neutrophils can be also involved in this formulation, and you can use some platelet-derived particles to eliminate bacteria. Again, another scheme just for you to memorize this. So how to obtain a platelet? You just add some calcium chloride to the suspension of platelets, following activation of these platelets, and you will obtain some platelet-derived gel. And this gel, for example, can be used for wound healing. In case you don't want to activate these platelets, you still can use their membranes to load some drug inside these membranes, or activate them with antibodies, and, for example, targeting the tumor cells. But all of this is very simple approach, to be honest, because it's just a membrane. What's so special about membrane? It's very simple to obtain such structures, but there are some structures exist that are very, very complex. And for example, this one is a platelet membrane covered nanomotors. So this is a state of the art in synthesis of different particles, because first you need to make some, if you look on this number B under letter E, you will see that this is template mediated fabrication of spiral particles, which are then covered in platelet membranes. So this is an uncovered spirals, and this is covered spirals. And if you add some additional staining for the membrane, you will see that this spiral entirely covered in platelet membrane. Why you need such a complex formulation as spiral coated in platelet membranes? Well, because you can use them as a sorbent for bacteria. So as you can see, nanomicromotors for diagnosis and therapy of infectious diseases. So platelets have natural ability to bind some bacteria, and if you have some spirals coated in platelet membrane, these spirals have again natural ability, maybe not very natural, but still they have ability to bind some bacteria on their surface. So as you can see, this is a bacteria on the surface of this platelet covered nanospirals. And these nanospirals also can move, as you can see here. So it's track of these nanospirals, nanomotors, which are moving in media and absorbing bacteria on their surface. So on fluorescent imaging, you can see you can see a spiral covered in blue-colored bacteria, and fluorescent intensity here showing that these motors really can absorb some bacteria from a media, even from a blood. As far as I remember, this is from some in vivo experiments. As it seems for me, this mostly looks like a toy still, because it's just a very nice, like, state-of-the-art formulation, some spiral coated in platelet membrane, and I do not believe that this is really can be implemented to eliminate some bacteria from our blood, because it will be very hard to eliminate these nanomotors from our blood, from our liver, kidney, and so on. Well, maybe in some application it will find some application in some fields, but not in bioapplication, medical application. And the last one, I want to show you another example of synthesis of organic particles. But important thing to note: these particles, which were coated in membranes of living cells, they will definitely possess a very low toxic profiles. So since they are looking almost like platelets, there will be not so much elimination of them from blood by, for example, macrophages. So biocompatibility of these particles can be really increased using this technique. But since this technique is very complex, let's say, as far as I heard, no one using platelet membrane coverage to lower cytotoxicity. So it's just too complicated to use it for such a very easy approach just for lowering cytotoxicity. For some targeting approach, for some delivery approach, yeah, membrane is good, but just to lower cytotoxicity, you just can cover your particles in lipids, in PEG or in PLJ, but covering in membranes, it's not the best option. It's just too hard to perform this. And the last one is the use of exosomes. Exosomes is another kind of biomimetic particles, and in case of exosomes, what is exosomes? If your cell uptakes something from a culture media, for example, I don't know, particles or just proteins or something like this, all of this will travel to early endosomes, and further it will travel to late endosomes, undergo some dissolution, and everything which is not essential for a cell will excrete to a culture media or other media, and this small formulation called exosomes. So it's something that came out of a cell. And these exosomes, they present in every culture media. They are just too small for you to observe with your eye and with the optical microscope. But there's a lot of exosomes in any of the culture medias, and these exosomes usually consist of cell membrane with different receptor on its surface, and some cells used exosomes for intercellular transport of different molecules. And this one is a very nice work. As you can see, it's Nature Communication, just a recent previous year. And what this scientists performed, so they take some dox, it's an anti-cancer drug, doxorubicin, and they use this drug. They treat some cells with enormously large amount of this drug. So a lot of drug pass through the cell, and they obtain dox-loaded exosomes. But on the surface of these exosomes, there still be some cancer receptors, and next they inject these exosomes in mice, and since well, the mice with a tumor, and tumor the same tumor as these cells. And so they, using this technique, they achieved a very precise targeting of these exosomes in this tumor. So it's very nice looking approach. And if you look on this, for example, TEM image, you can see how this exosomes looks. They are very small, basically. It's like 20 nanometers in size. And more important, it was just not exosomes. It was also some attempts to produce exosome-covered magnetic nanoparticles. So as you can see, it's some kind of core shell, and this is a very nice approach to shield your particles from immune system. So it's just exosome. There's no inside, but inside is a magnetic core. And if you look on this survival rate plot, you can see that these exosomes are very efficient in tumor suppression, because they have the same suppression efficiency as high concentration of DOX. So instead of using a very high, obviously toxic concentration of DOX, you just can use exosome loaded with DOX and achieve the same survival rate as with high concentration. Compared to just usual concentration of DOX, survival rate increase maybe twice. So it's a very nice result. And these particles obviously will be quite biocompatible, because they are formed from a cell membrane. Yes, cancer cell membrane, but still it's a cell membrane. And there's a lot of exosomes in our blood, so it's just a natural component of our organism. And this approach is quite biocompatible. Well, it was the last slide, I believe. So if you have some questions, I will be happy to answer to these questions, and I will just wait a couple of, maybe 1 minute for you to formulate your question if you have some. Okay. If you have no questions, I need to pick up people to present their topics on next Friday. So again, next Friday it will be not that easy to find a schedule. Okay, so next Friday after lecture 4, four of you should present your topics. Again, topics, let me find the topic list here. Some of you already have their some topics, but not all, because not everyone filled what they need to fill. And I believe Nastya and Alexander want to give their presentation next Friday. Yes, so 1, 2, but we need additionally 2 people who want to volunteer. So last chance to volunteer, or I will pick randomly. Ten seconds to think. No one, yes? Just now it will be fatality, yes? 1, 2, 3, 14. Brink. It will be a pure random. So first two to present. 3 and 14. 3, it's up. 14 already, okay. Once more time again. It's not that easy. So without 3, without 14, and without 9. First one to present, 7. Okay. So let me just fill with a color. Next Friday. Next Friday. Next Friday. Next Friday. That's all for today. If you still have no questions, can you present on 13? There will be no lecture on 13, so it will be like 11, 16 of December. Again, presentation 5 minutes long, and your report, your text, you should prepare some text. 5 lists, no more than 5 lists. So less than 5 is you can present, make less than 5 lists, but no more than 5 lists. 5-minute presentation, 5 lists long report. So again, presentation will be next Friday, 4 people. Tuesday 8th will be 4 people. Friday 11th will be 4 people, and the last day will be Wednesday 16th, 4 people. So there, I believe there's some, well, it will be like 12 people, yes. Okay, we will manage this, but again, 4 people next Friday. Okay, if there's no questions, if there's any question, you can always ask me personally via email. This file can be found in common Google Drive folder, which was sent to you along with different materials with the course. So it's on a Google Drive in a common folder called Nanotoxicology course 2019. So well, here. So on Google Drive, Nanotoxicology course, student reports topics. You open this, you need to fill object of research, and I will fill a report topic for you. So if everything is clear, I hope at least that everything is clear. Okay, if you have any questions, again, you can contact me via email or WhatsApp personally or something, or other ways you find suitable. That's all then for today. See you next Tuesday, December the 1st. There will be a test in the middle of the lecture to assess your knowledge of lecture 2, today's lecture, so do not miss it. And there will be no presentation next Tuesday. Presentation will be next Friday in a week. Okay. So see you next lecture.